The overall goal of this video is to introduce a technique for differentiating human pluripotent stem cells into cardiomyocytes, and to demonstrate the use of these cells for studying myocardial repair after an ischemia-reperfusion injury.
This method can help answer key questions about engineering heart tissue with human iPS cell-derived cardiac cells for myocardial repair. The main advantage of this protocol is that it allows scientists to collect purified cardiomyocytes, smooth muscle cells, and endothelial cells, and generate heart tissue with these lineage of cells. Ling is a postdoc in Dr. Zhang lab in UAB, and Greg Walcott is a cardiologist in UAB Hospital. Trina is an MRI specialist in UAB Hospital.
To induce pluripotent stem cells into cardiomyocytes, first, coat the wells of a 6-well plate with growth factor-reduced gelatinous protein mixture cooled to 4 degrees Celsius. Then, refrigerate the plate overnight at 4 degrees Celsius. The next day, aspirate the gelatinous protein mixture and seed the plates with 100 million stem cells per well. Never keep the stem cells out of the incubator for more than 15 minutes. Also, always use fresh media. In this case, use MTESR1 medium supplemented with 10 micromolar of ROCK inhibitor. Then, culture the cells with 5% carbon dioxide at 37 degrees Celsius and refresh the medium daily by gently aspirating away the old medium without touching the cells